mic on, correct?
Correct.
Okay. All right. Thanks everyone for coming out to watch this today. And I'm going to present some work I did with Carlos Pires and Dr. Rice this summer on the short-term soil health impacts from diversification and intensification in rain-fed cropping systems. So the motivation for this work was due to the lagging production in the Southern Great Plains region, which is derived in part from declining soil health, as well as in conjunction with factors from climate change. So in order to create a more resilient agricultural system and increase soil health in this region, we need to increase the efficiency, the resource use efficiency, the diversity, and the intensity of the cropping system. Since I'm going to talk a lot about intensity to kind of define that term more for this poster, it's just dealing with like the frequency of the cropping. So how many crops per season would be a way to think about it. And our hypothesis was, going off of previous work, was that an increase in intensity would lead to an increase in soil microbial biomass and enzyme activity, which are two common measurements for soil health broadly. Moving on to the methods, we chose a subset of 4 treatments from the rain plots at Ashland Bottoms. Looking at this figure over here, starting with the control, that is the wheat fallow rotation. And then that's kind of the baseline for what the region is doing currently. Then looking at the adaptive forage treatment, that's wheat fallow, and then currently this summer there's a forage, and that's a wheat forage for the summer. The intensive treatment is wheat, and it had sorghum last summer, and then another wheat, and then it had soybean. And then the dual purpose semi-intensive has wheat fallow and then triticale instead of wheat, and then currently is in soybean. So the main differences to pick up here are that the sorghum had this over the summer, and then we had triticale instead of wheat for the dual purpose, or the intensive had sorghum last summer and the dual purpose had triticale. The others note that there shouldn't be like too much different because a lot of the differences are just starting to come in this summer. And this blue or purple line indicates our sampling point. So going forward into the methods we used, we looked at a PLFA, which is it uses phospholipid fatty acid biomarkers in order to develop a sort of coarse taxonomic identification, and you can get an estimate of the microbial biomass for each of those groups. So you can get like fungi and then break fungi into like saprophytic versus AMF, and then bacteria gram negative, gram positive, and actinomycete. You can't get too much more specific than those groups, but those still yield a lot of important information. Then going on to soil enzymes, we used a colorimetric method, which is important to note here because it's different from the one Shelby used and she'll present in a later presentation. And the colorimetric method is a more standardized procedure that's used by NRCS for soil health analysis. And what the soil enzymes do is give a sense of the potential microbial activity in the soil. And it doesn't say maybe like what they're exactly doing, but it's more what the potential in that soil is. So we looked at 4 different enzymes. Beta-glucosidase deals with the carbon cycle. The NAG, as I'll call it here, deals with carbon and nitrogen cycling. Acid phosphatase, phosphorus cycling, and aryl sulfatase with the sulfur cycle. All of these are hydrolytic enzymes, and they deal with rate limiting steps in these cycles. So importantly, later on, the beta-glucosidase and the aryl sulfatase are less likely to be interfered with any fertilizer treatments, whereas the acid phosphatase and NAG are more likely to be fertilizer applications impact them more strongly. Moving on to the results, we collected both 0 to 5 and 5 to 15 centimeter soil samples from these plots this summer, but I'm only presenting the 0 to 5 centimeter data due to time. But there were less differences seen in the 5 to 15, although they were different and interesting. So looking firstly at the PLFA results, chart A here is the total microbial biomass, and chart B is the total bacteria. So to point out the differences here, since they might be hard to see on the screen, the adaptive forage and the semi-intensive are statistically equal. The control and the semi-intensive are also statistically equal, and the intensive and the control are statistically equal as well. Notably, the intensive and control, so the intensive is lower than the adaptive forage and semi-intensive, even though it's equal to the control. So that was a little confusing, but essentially like the intensive is the lower value here, and it's equal to the control. Moving over to the bacterial biomass in B, the adaptive forage and semi-intensive were again equal, and the control was equal to them as well. And similarly, the control was equal to the intensive, but the intensive was different from the semi-intensive and adaptive forage. So the main takeaway from these two charts here is that the intensive is tending to be lower than the other two treatments and pretty close to the control, although numerically anyway, it's trending lower. The other two are trending a little higher. So moving on to the enzyme data, this is much more clear, although there are a couple outliers as can be seen in A. So A is the beta-glucosidase. And again, this was one that's not impacted by fertilizer applications as much. And you can see here that all the adaptive forage, control, and semi-intensive treatments are equal in beta-glucosidase activity, whereas the intensive is statistically different. And the same thing with the aryl sulfatase to the left. So these are some nice trends, and the big takeaway here is, again, that intensive treatment is actually trending to be lower. So moving on to conclusions from those charts and from some of the other analysis we did, it looks like the intensive crop rotations are actually tending to decrease the soil biological properties in the short term, which is the opposite of what we expected. And the other treatments, going mostly off this PLFA data, are sort of trending to be higher, although that didn't come out in the statistics as much. So we think that this intensive treatment that's going kind of the opposite as we expected was primarily due to moisture. And from last summer, the sorghum taking up a lot of that moisture, and that actually being what's limiting the microbes in this context, even though we predict there's going to be higher carbon inputs from that sorghum residue and roots, but we think that in this case, what's actually limiting is the moisture for that microbial biomass and activity, which is a pretty interesting and unexpected result. Since this is just a short-term analysis, the future research is going to be super important here and to build off this. So looking at the seasonal variability for biological properties is going to be important, as we also looked at some baseline PLFA samples from 2019. And what we saw when comparing those to the 2021 is that there were some differences, but when we realized that the sampling time then was around December, late November, December, and the ones we took were in the spring for 2021, we realized that maybe comparing them wouldn't be very informative because of the seasonal variability due to like crop phenology and things like that. So And we need to really create a standardized method for sampling these plots in order to compare biological properties over time. Moving on, we're also super interested in carbon dynamics. Although we weren't able to get the samples back in time to look at any differences that might have occurred in carbon for these for this poster, it's going to be interesting to see how the microbial properties and the carbon develop with these different treatments, because carbon's really a big, focus of soil health. It's like the driver of a lot of soil health. So, and that'll also deal with the soil physical property changes we were thinking might occur in the intensive treatment as well. And then we also need to further advance our knowledge looking at the soil moisture and the possible decreases we saw with the intensive treatment due to that, and kind of test that hypothesis and see if that's a correct analysis. And then going forward, just thinking about, this is a short term, this poster is focused on the short term, and then like understanding how these treatments would change over time, and how intensification and diversification are going, in different systems are going to change over time, but also understanding having the short-term impacts, so we can understand maybe people, you'll have early declines in microbial activity, but over the long run, things will increase. So understanding all those will be super important. So thanks to the Soil Microbial Agroecology Lab for all their help, and Carlos for all his help as well, the Kansas Department of Agronomy, and then the Rainfed Agriculture Innovation Network. Any questions?